Okay, so today I'm going to deal with the nervous tissue. Remember that at the beginning we mentioned that there are 4 types of tissues in the body. We dealt with the epithelial tissue, we dealt with the connective tissue, and then the muscular tissue was dealt with last week, and today I'm going to deal with the nervous tissue. So the nervous tissue, it controls and integrates all the activities in the body together with the endocrine system. That's why we're going to study them in one block. And it has these 3 basic functions, which are actually you can compare these functions to the function of a computer system. For example, like here, the basic function is sensory, sensing changes with sensory receptors. It's like input to the computer, and then integration, integrating, interpreting, and remembering these changes in the environment. It's like the central processing unit, and which provides for integration. And then the third function is the motor function, where it reacts to the changes by acting on effectors. Either these effectors could be muscles or they could be glands. So this is like the output of the computer. So input, processing, and output, sensory, integration, and motor. These are the functions of the nervous tissue. The nervous system is divided into central nervous system and peripheral nervous system. The central nervous system, this is a structural division. It's like structural, anatomical division. So the central nervous system consists of the brain and the spinal cord, and then the peripheral nervous system, which connects the central nervous system to the peripheral structures, consists of the nerves that are connected to the brain, so they are called cranial nerves. We're going to study these cranial nerves in the next few lectures. There are 12 pairs of these cranial nerves. And then there are nerves connected to the spinal cord, so they are called spinal nerves. And again, we are going to study them. There are 31 pairs of these spinal nerves. So it's central, brain, spinal cord, peripheral, which consists of the cranial nerves and the spinal nerves. And these, they connect the central nervous system either to the receptors, where they bring sensory information, or they connect the central nervous system to the effector organs, muscles, and glands. Functionally speaking, the peripheral nervous system can be divided into somatic and autonomic. The somatic is the voluntary nervous system, and here it's either sensory or it's either motor. Motor, it supplies the skeletal muscles, because these are the voluntary muscles. It is under our will. And then the sensory is, it brings sensations from receptors, like for example, we call it somatic sensations, like receptors on skin, like pain, touch, and temperature. These are special kinds of sensations called somatic sensations. If you compare this with the autonomic nervous system, which is involuntary, the autonomic nervous system, again, it has motor output to muscles and to glands. But the muscles are not the voluntary muscles, are not the skeletal muscles. It supplies smooth muscle fibers, it supplies cardiac muscle fibers. That is these are the involuntary muscles that are supplied by the autonomic nervous system. And this does not mean that the autonomic only carries motor fibers, but also carries sensory fibers. But sensory fibers, which are not somatic, they are visceral fibers, visceral sensation. Like when you have, for example, like let's say acidity in the stomach, so you will feel like burning pain in the stomach. This is not a type of pain that you feel it on your skin, or for example, when there is spasm in the intestines, which results in like feeling of gases and spasm because of the These sensations, they arise from viscera. They differ from the sensation of pain, touch, and temperature. Like, let's say, if somebody can touch your intestine, then you will not feel that touch because there are no somatic sensory fibers supplying your gut. So they are not sensitive to touch, but they are sensitive, for example, to distension. If they are distended with air, then you will feel that sensation. So that is the kind of sensation that is carried by the autonomic nervous system. This autonomic nervous system can be subdivided into sympathetic, parasympathetic, and enteric. The sympathetic, we're going to talk about details about these later on, but this is just an introduction. The sympathetic division prepares the body for flight or fight. So, for example, it increases the pulse rate, contractility of the heart, increases the blood supply of muscles so that you can prepare your body for fight or flight. But the parasympathetic prepares the body for what we call rest and digest. So it increases peristalsis, it increases the secretions, gastric secretions, and so on. And there is an enteric division. This enteric division is a kind of a local circuit which is present only in the gut and controls the local environment of the gut without the need to be connected or to receive orders from the central nervous system. It can work independently on its own, receiving information from local environment and providing orders for the local environment. And this is only present in the gut, and so it's called the enteric division of the autonomic nervous system. Some of these links that are provided in the lecture were not working. If you downloaded the lecture before yesterday night, then some of these links are not working. So, and then I corrected them. So if you download it now, these links are working. Now, histologically speaking, the nervous system consists of two types of cells, neurons and neuroglia. So I will start with the neuroglial cells. The neuroglial cells, as you can see, the name means that they are nerve glue. So they were thought earlier when they were discovered that they act like a connective tissue for the nervous system as a glue, only supporting. This is one of their main functions, but then later on they discovered that there are different types of these glial cells and they have different functions, and their function is not restricted to being a glue or connective tissue or supporting tissue for the nervous system. They constitute about half the volume of the central nervous system, but because they are very small cells, as you see them here, if you compare them to the neurons, they are very small, and so they are much more numerous than the neurons, about 50 times more numerous than the neurons. They differ from neurons in that they can multiply. Neurons cannot multiply, but these cells, they can divide, multiply. That's why tumors arising from these cells are very dangerous, because these cells can multiply. The tumor, which is called glioma, can increase in size rapidly. There are many types of these cells, as I mentioned, because they are not only a glue, so they have different functions. There are 4 types in the central nervous system and 2 types in the peripheral nervous system. In the central nervous system, we have the astrocytes, oligodendrocytes, microglia, and ependymal cells. In the peripheral nervous system, we have Schwann cells and the satellite cells. We are going to study these cells separately now. So the astrocytes, which are present in the central nervous system, as the name indicates, astro means star. So they are star-shaped cells, astrocytes. Cyte is a cell. So they are star-shaped cells, and as you can see them here, they have processes that are connected to blood vessels. Here, the blood vessels are lined with endothelial cells, which are simple squamous epithelium cells, which have tight junctions in between them, and they produce what we call the blood-brain barrier, BBB. So this blood-brain barrier is mainly because of the tight junctions between endothelial cells, but these cells, the astrocytes, as well, participate in the formation of this blood-brain barrier. As the name indicates, it acts as a barrier between the blood and the tissue of the brain, the extracellular compartment of the brain. Because sometimes there are fluctuations in the constituents of the blood, and these fluctuations will be reflected on the constituents of the extracellular tissue. So the presence of this blood-brain barrier will control and prevent these fluctuations in the extracellular tissue of the brain, because the brain is so delicate and cannot withstand much changes in the extracellular tissue. Also, they act as a barrier for toxins and certain drugs. So it's very important for the function of the brain. These cells participate in the formation of this barrier. Of course, we should always remember that they provide structural support, and they, as I said, they regulate the balance of the ions and which are present in the extracellular matrix, and they metabolize neurotransmitters. The other cells are called oligodendrocytes, and so these oligo means small number because they have small number of processes, so they are called oligodendrocytes, and these cells are responsible for the formation of the myelin sheath in the central nervous system. You can see them here. Which one of them? It's the red one there. They have this one here. They have processes, and these processes, they wrap around the axons of the neurons in the central nervous system, and this wrapping around the axons is the production of the myelin sheath. Yes?
Is it possible for you to just move the slide down so we can see it?
I don't know how to put it down.
No.
Yeah, sorry. Yeah. I think it's already set. I cannot move the projector. Yeah. So, so they are oligodendrocytes. You can see them here. You can see the name here. Oligodendrocyte is the same as this one. So they have processes that wrap around and produce the myelin sheath. We are going to talk about the myelin sheath in a moment. But this myelin sheath, just to give you an introduction, it's a sheath that is made of multiple wrappings of cell membrane. So it's the cell that moves around the axon, wraps the axon, and so it acts as an insulator. It's like, let's say, the plastic material that's put around the electrical cable. So it acts like an insulator, separating that cable, that axon, from the nearby axon. So here in the central nervous system, this insulator is provided by the oligodendrocytes. In the peripheral nervous system, we have Schwann cells, which produce this insulator in the periphery. So they are analogous to the Schwann cells of the peripheral nervous system. Now the third type of neuroglial cells in the central nervous system are the microglia, and as their name indicates, micro, they are small. So they are small glial cells. They are located near blood vessels, and these are phagocytic cells. Actually, embryologically speaking, they do not develop from the ectoderm, which provides the development of the nervous tissue. They develop from mesoderm. They develop from bone marrow cells that provide macrophages and monocytes, which are the cells that engulf foreign substances. And this is their function in the central nervous system. We can say that they are the macrophages of the central nervous system. They have phagocytic role, and in addition to the supporting role. The other cells are the ependymal cells, and these cells, as you can see them here, they line the cavities which are present in the central nervous system. We are going to talk about these cavities later on. In the brain, in the spinal cord, there are cavities they contain fluid, which is called cerebrospinal fluid, because it's present in the cerebrum, which is the brain, part of the brain, and present in the spinal cord. So cerebrospinal fluid, this cerebrospinal fluid is produced by these cells that line the cavities that contain the cerebrospinal fluid. This cerebrospinal fluid has many functions. We are going to talk about them later on. It provides nutrients, it washes out metabolites, it makes the brain, supports the brain because it keeps the brain floating in water. So we're going to talk about this cerebrospinal fluid later on. It is these cells that produce the cerebrospinal fluid. So these are now 4 types of glial cells in the central nervous system. We have the astrocytes, the star-shaped cells, the oligodendrocytes that produce the myelin sheath, the microglia that engulf, and the ependymal cells that produce the cerebrospinal fluid. In the peripheral nervous system, we have 2 types of cells. So we have the satellite cells, which are located here. This is a neuron. You see, this is the cell body of a neuron. It's a very, very big cell. We mentioned that the microglia are much smaller, the neuroglia in general are much smaller than the neurons. So they are very small cells as satellites around the neuron, and they are present in ganglia. A ganglion, we're going to talk about ganglia later on, but a ganglion by definition is a collection of nerve cell bodies outside the central nervous system. In the central nervous system, we have collections of nerve cell bodies which constitute what we call the gray matter of the central nervous system. And the white matter, the surrounding white matter of the central nervous system is formed by the axons that are surrounded by the myelin sheath, because this myelin sheath is mainly formed of fat, so it looks white. So there's white matter and gray matter. In the peripheral nervous system, there are places where there are collections of nerve cell bodies, and these constitute the ganglia. And around these neurons in the ganglia, there are supporting cells which are called satellite cells. The Schwann cell, as you can see it here, it is the it corresponds to the oligodendrocyte. But here, as you can see, that the Schwann cell, one cell wraps itself around one nerve fiber, one axon. While in the oligodendrocyte has several processes, and these processes, each of them makes a myelin sheath of an axon. So one cell provides the myelin sheath of multiple axons in the central nervous system, while in the peripheral nervous system, the Schwann cell, the analog of the oligodendrocyte, provides the myelin sheath of one axon only. I will talk a little bit more about myelination process, but let's talk about the neurons now. We said that there are 2 types of cells in the nervous system, neurons and neuroglia. Neuroglia are 4 in the central nervous system, 2 in the peripheral nervous system. Now I'm going to talk about the neurons. The neuron is the functional unit of the nervous system. They are the excitable cells. They are characterized by the ability to transmit electrical impulses, unlike the neuroglia. Neuroglia are the glue. And each cell has a cell body, as you can see it here, a big cell body, and processes. The cell body has a nucleus with a very prominent nucleolus. Remember, the nucleolus, the site of production of ribosomal RNA. So this gives a clue that this cell is involved in protein synthesis. Also, you can see that there are some granules, like some bodies in the cytoplasm, which are called Nissl bodies, and these are can be easily stained. They are present in the cytoplasm of the cell. They extend into the processes, into the dendrites, and these represent a rough endoplasmic reticulum and free ribosomes. Again, the presence of rough endoplasmic reticulum gives a clue that there is protein synthesis, because proteins are synthesized on the rough endoplasmic reticulum. In addition to that, there are other components like mitochondria, like the neurofilaments, which provide for the cell shape, and microtubules. The other features of a cell are also present. But the prominent feature here is the presence of Nissl bodies, which is rough endoplasmic reticulum and free ribosomes, the presence of a prominent nucleolus in the nucleus. The processes are two types. They are either dendrites or an axon. So these are, this is a motor neuron seen in a light microscope, and this is an electron microscope view of one of the neurons as well. The neurons, they vary in their shape, they vary in their size, so it's not like they are not fixed. Not only in their function, but also they differ in their shape. So the dendrites, these are multiple processes and they are branching. Usually they are multiple, branching and short. If you compare them with the axon, which is a single, long, these are multiple, short, and branching, and they represent the receptive zone of the cell. They receive the impulse, and they have specialized surfaces of contact, as you can see them here, called spines, dendritic spines, special areas for contact. They communicate or contact with other neurons. You will see that in a moment, how these junctions are formed. And they contain, as I mentioned, they contain Nissl bodies as well. So Nissl bodies are not only confined to the body of the cell, but also they extend into the dendrites. The axon, on the other hand, conduct impulse away from the cell. You can see the direction of the arrow here, so away from the cell, while these, they conduct impulses toward the cell. So these are receptive, and this one is the conductive zone. It is long, thin, and cylindrical, and when it's connected to the body, the part of the body to which it is connected, as you can see it here, is cone-shaped, called axon hillock. Hillock means a small hill. So this part of the cell body is called the axon hillock, and it connects to the axon at a place which is called the trigger zone. Here, this is the trigger zone. This is where the impulse, the electrical impulse, starts, the trigger zone. It's between the initial segment of the axon and the axon hillock. We call it the trigger zone, the beginning of the electrical signal. Many axons, they have branches, which are called collateral branches, and the end of the axon will also branch. These are not collateral, these are terminal branches or axon terminals, but these are the branches, a couple of, or maybe more than these, but not like the dense branches that are present in the dendrite. There are a few of these branches, collaterals, and at the end, the axon divides into the axon terminals, and these are, the far end is swollen, and they form what we call the end bulbs or synaptic botons. And these are filled with neurotransmitter. Remember, we mentioned something about them in the neuromuscular junction. So there are synaptic vesicles at the distended ends of the neurons. Now, so here you can see the axon is covered by myelin sheath here. This is in the peripheral nervous system. So this is the cell is a Schwann cell, and the cell will wrap itself around the axon several times, probably 100 times. It rotates around the axon, and this happens during fetal life, and after birth, the process of myelination continues. The myelin becomes thicker and thicker, and so here you have multiple layers of cell membrane. So when the cell wraps itself around, the cytoplasm will be squeezed out. So the cytoplasm and the nucleus will be in the periphery, and the myelin itself is only formed of multiple layers of cell membrane. Remember, the cell membrane it's a lipid bilayer with some proteins like islands or icebergs. So that's why the myelin, therefore, is multiple wraps of cell membrane, and it's a lipoprotein. It's mainly lipid material. That's why it looks white. And the areas that contain myelinated fibers, they are whitish in color. And especially in the central nervous system, they form the white matter. In other neurons which are not myelinated, like this one, you can see here that there is one Schwann cell is forming an insulator around several axons, but they are not wrapping themselves around the axon. So they are just providing an insulator, but not wrapping around. This is one cell around one axon, so the fiber therefore will become thicker, but these will be, the nerve fibers are thin, and it's one cell wrapping itself around several axons, and so it's called unmyelinated. So the unmyelinated, they do not have myelin sheath, but still they are insulated because they are covered by a Schwann cell. But a Schwann cell covers more than one axon, and it doesn't wrap itself around the axon like in a myelinated nerve. And here you can see that multiple Schwann cells are present in sequence on the axon, and there is a space in between them, which is called the node of Ranvier, where the axon is bare, is not covered by myelin, space here, the node of Ranvier. And so you can expect that the electrical impulse will jump, leaping from one node of Ranvier to another node of Ranvier. So instead of the impulse, the electrical impulse moving throughout the entire length of the cell membrane, which will take it longer time because it has to move from one region to another gradually. Here, the impulse is jumping from one node to another. That's why these neurons, the myelinated axons, they are rapidly conducting. If you compare them with the unmyelinated axons, the unmyelinated axons are usually thin, and they are, or unmyelinated nerves are usually thin, and they are slow conducting because the impulse has to pass gradually along the entire length of the membrane. But in the myelinated, it can jump between the nodes of Ranvier, so they will be rapidly conducting. Okay? These are present in the, of course, in the central nervous system. I would like to remind you again, it is not a Schwann cell that produces the myelin, it is the oligodendrocyte that produces the myelin. And these myelinated nerves in the central nervous system will form the white matter, while the gray matter contains cell bodies and they contain unmyelinated fibers. This is how the myelination process is taking place during embryonic life, and then after birth until adulthood. So you can see the cell is wrapping itself, multiple wraps, sometimes 100 wraps around the neuron. So that's why these neurons, even if they are myelinated, but they are not that much myelinated in infants. That's why the reflexes or the movements of the infants are clumsy, they are slow if you compare them with the reflexes of the adults, because in the adults the neurons are highly myelinated, so they are rapidly conducting. This is how a synapse is formed. The end bulb of the axon here, it's called presynaptic end, and this is the postsynaptic neuron. This is how two neurons are connected to each other. So actually, it is not an actual connection, it is not a physical connection. So the electrical signal cannot pass directly from one neuron to another. It is there's a gap here, which is a synaptic cleft, it is called, and that's why we need chemicals, which are called neurotransmitters. So this is not an electrical conduction, it is a chemical conduction. And the end bulb has synaptic vesicles, and these synaptic vesicles, they contain neurotransmitter. They are secretory vesicles, in fact, and when the impulse reaches the end bulb, these secretory vesicles approach the cell membrane and open here, release the neurotransmitter. The neurotransmitter will go to the postsynaptic membrane, and there are special receptors and then this will trigger the postsynaptic neuron to start an action potential. So the electrical signal is not directly conducted from one neuron to another. There is a gap, and that's why we need a neurotransmitter. It's same as in the neuromuscular junction. In the neuromuscular junction, there is a neuron and a muscle, and the neurotransmitter is acetylcholine. Here there is a neuron and another neuron, and the neurotransmitters, there are different types of neurotransmitters, not confined to acetylcholine. This diagram is showing you how the impulse is conducted in a myelinated neuron. You can see here the impulse is jumping, leaping from one node of Ranvier to another node of Ranvier. So that's why it will be faster. These neurons, the myelinated neurons, are faster conducting than the unmyelinated neurons. Structurally, these are different types of neurons. As I mentioned, they are not all the same. So this one is called multipolar neuron. There are multiple dendrites, a single axon. Actually, most of the neurons are multipolar neurons, but there are some other neurons that are bipolar, like this one. So they have two processes. One of them represents a dendrite, and the other represents an axon. Okay, so they are bipolar. These types of neurons are present in the retina, for example, in the organ of Corti in the ear. We're going to study that later on, in the olfactory epithelium. So they are more or less related to special sensations. The other type of neuron is called unipolar, only one process that immediately divides into two processes, like a capital letter T. That's why sometimes the other name of these neurons, they are called pseudo-unipolar neurons, because they are not exactly unipolar. It's the pole, the process, immediately divides into two processes. So you can say that they are unipolar or pseudo-unipolar neurons. And these processes, one of them is connected peripherally and the other connected centrally. So the one that is connected peripherally acts like a dendrite. It brings impulses toward the body, and the other one acts like an axon, the central process. It takes the impulses away from the body. These kinds of neurons are especially present in what we call dorsal root ganglion cells. We're going to talk about dorsal root ganglion cells. They are sensory neurons. They send processes, for example, to the skin. And so when somebody touches your skin, you will feel the touch. This is transmitted by the peripheral process. The neuron is present in a ganglion outside the central nervous system, and then the impulse will continue through the central process to reach the central nervous system so that you can feel the touch. So these are the three main types of neurons, the multipolar, bipolar, and uni or pseudo-unipolar neurons. Functionally speaking, neurons are either sensory, as we mentioned, or motor. Usually we use the word afferent in the nervous system, which means sensory, and efferent for the motor. Yet there are many other neurons, as you can see them here, which are located between the motor and sensory neurons, and these are called interneurons. So these are interneurons. They are either association or they are projection. Like, for example, projection neurons that they connect one part of the central nervous system, higher part to a lower part, or a lower part to a higher part. Or they are just association neurons. This is like a reflex arc, as you can see. So, for example, if you feel something hot, let's say, then the temperature sensation will be transmitted through a sensory neuron. This sensory neuron is a kind of pseudo-unipolar neuron. It has cell body in a ganglion outside the central nervous system, and that is its peripheral process. Here is the central process, and then this synapse with an interneuron, and then the interneuron will carry the impulse to a motor neuron. Motor neurons are located in the ventral horn of the spinal cord, and the motor neuron is a kind of multipolar neuron, this one. So it has several dendrites, short processes here, but there is only one long process that will take the impulse to the muscle, and so you will withdraw your hand from the hot object. Okay? So this is how functionally neurons are classified. And of course, the neurons, they produce circuits. Like it's not all the time. Actually, I don't think that there is one-to-one contact between neurons as we might sometimes simplify them. Like I have demonstrated here. It looks like it's one neuron, then one sensory, then one interneuron, and then one motor neuron. This is not the actual situation, because when you need to withdraw your hand, then you need several neurons to act, to work on different muscles and different nerves to produce this movement. So that's why we need what we call a neuronal circuit. Neuronal circuits. These are of different types. The study of these circuits is in one way or another also related to computer science. So some of these circuits are like diverging, like this one. One neuron affects multiple neurons, and these multiple neurons affect multiple neurons, so there will be an amplification of the signal. Or it will be converging circuit. So multiple neurons affecting one neuron only. Or the other one is called reverberating. So reverberating, we see that the earlier neurons they will receive feedback or will be affected by later neurons. Because you can see here that this is an axon of a later neuron, and this is its collateral. Remember, we mentioned that axons have collaterals. So this is a collateral. So the collateral of the axon will bring like feedback which might inhibit or stimulate, because the impulse or the signal is not always stimulating. Sometimes it's stimulating, sometimes it's inhibiting. So for example here, like this cell, which receives impulses from many other cells, some of them are stimulating, others are inhibiting, and the action that will be taken by the receiving cell depends on the balance of the inhibiting and the stimulating impulses, which of them will predominate. And then the other one is parallel after discharge. So you can see here, like a single cell stimulates group of cells, and all these cells will stimulate or will act on whether stimulating or inhibiting one other cell. So these are different types of neuronal circuits. It's not a simple one-to-one relation between the neurons. That's why the cells, there are multiple dendrites, and these dendrites, they are connected to many other cells, many other neurons, and even the axon has multiple processes at the end, so it's not like one-to-one relation. There are circuits in here. Now I will deal with the regeneration and repair. We should keep in mind that in the nervous system there is a lot of plasticity in the nervous system, but there is limited regeneration. As I mentioned, the neurons, they do not multiply at all. What multiplies in the central nervous system are the glial cells. But the neurons, they can repair themselves. And this repair is limited to the peripheral nervous system and not to the central nervous system. But whether central nervous system or peripheral nervous system, neurons have plasticity. This is something different from regeneration or repair. Plasticity means that throughout life, the neurons, they sprout new dendrites, make new connections, synthesize new proteins, so make new synapses, new connections. This is what we call it plasticity. But there is limited ability to regenerate, and as I mentioned, that regeneration or repair is for the damaged dendrites or axons in the peripheral nervous system. but not in the central nervous system. It is still under investigation. Now there are many experiments about repair in the central nervous system, but it is still under investigation. So it is very little and still under investigation. We can say that repair is not officially possible in the central nervous system, but it is possible in the peripheral nervous system. How does this repair take place in the central nervous system? In the peripheral nervous system, again, there are rules for that. The rule is that the cell body should be maintained. It's only the axon or the dendrite is cut. But if the cell body is destroyed, then no repair will take place. And the second thing is that Schwann cells, because we are talking about peripheral nervous system, the myelin, these myelin, still they can maintain their ability to divide and multiply, because they are neuroglial cells after all, so they can multiply. So they should retain their ability to multiply. The cell body should be still viable. The injury is either in the axon or in the dendrite. And the third rule here is that no scar tissue is present in between the cut nerve. So that's why when you have a nerve injury, for example, somebody cuts his forearm, well, this might not only result in a cut of skin and blood vessel, it might also involve a nerve. So that's why during surgery, they have to approximate the two ends of the nerve together. They have to suture them, approximate them together. Because if any fibrous tissue is formed in between the two ends, then the sprouts of the new axons cannot build and reach their destination. So that's why they should be approximated. No scar tissue should be formed at the cut end. So these are the rules for regeneration to take place. Again, I repeat that this is only possible in the peripheral nervous system at the moment, but not in the central nervous system. What happens briefly is that there is what we call chromatolysis in the first 1 or 2 days after the injury. So these Nissl bodies here will disintegrate, and then after that there will be what we call a Wallerian degeneration. The entire axon, the entire process distal to the site of injury will disintegrate, and there will be also proximally part of the process will disintegrate to the nearest node of Ranvier. Remember the nodes of Ranvier? And then after that, the Schwann cells, they will multiply and produce a tunnel or a canal through which the axon can sprout and pass through. So that's why we need these Schwann cells. One of the rules of regeneration here is that Schwann cells remain active and form a tube. This tube is necessary so that the remaining part of the axon can sprout and pass into the tube, and this tube will guide it to the effector organ, and this should not be blocked by fibrous tissue, as I mentioned. So that's why we have to approximate. And this is a very slow process, as you can see, that the axons, they grow and bud about 1.5 millimeter a day. 1.5 millimeter a day. So it takes long time to repair itself, but is still possible in the peripheral nervous system, not in the central nervous system, on the account that the cell body is still intact, Schwann cells can still multiply, and no scar tissue is formed. Okay? Questions? Thank you.
I'm still with final exams will be also online.
Yes. Everything, all exams are online.
Yeah. And then for the lab quiz, that one I'm not sure. So basically the materials that's gonna be in the lab quiz will be like in the virtual labs that in the Bright Cities.
Yes. Yeah, but for the they are not considered as an assignment, so you don't have to submit the answers to the questions, but they are there to give you an idea about what is
Gonna be in the lab.
yes.
The practice exam that is on, we can see on the website, is that just for the file, or is that useful for presentation?
It's the same, and also the questions that you have them after each virtual lab, they are the kind of questions that you expect in the practice. You're welcome.
Okay.